Hello and welcome to LaunchPod. I am Paul Stimpson, bringing you a new offering from us this month, and it's the second episode of It's Not Rocket Science, where we put the spotlight onto our researchers in residence and hear about their projects. On this episode, we'll be hearing from one of our very first researchers in residence, and they are very unique in the fact that they are two researchers working alongside each other on this. It's Beatrice and Salome from the University College London, who are working with CPI, the Centre for Process Innovation, part of the High Value Manufacturing Catapult. But before that, let me give you a quick introduction to the Innovation Launchpad Network. The Innovation Launchpad Network Plus, funded by the Engineering and Physical Sciences Research Council, the EPSRC, brings together leading universities, the Catapult Network, and regional innovation ecosystems, and enriches the exchange of ideas and knowledge across these organisations. One of the ways we do that is through the Researcher in Residence scheme, and this really is a flagship activity for us as a network. For those who don't know, we award up to 50k to researchers in the UK whose research proposal fits within at least one of our key themes, which are net zero, resilience, and healthcare and wellbeing. These projects are awarded on a collaborative basis, meaning that it is a requirement for researchers to work alongside at least one of the catapults. You can find out more about the catapults and other details about the scheme over at innovationlaunchpad.ac.uk. Taken from our Tranche 3 launch event that took place at the beginning of October this year, 2023, we asked one of our successful applicants from the first round just to give us a brief overview of their project and how they found the application process. And here's what they said.
So we've been asked to talk about our experience so far, so a bit about our project, what we're hoping to get out of the experience, and the process of applying, and a few points in our first week. So that's now slide 2.
So treatments based on nucleic acids have huge potential, and our mission is to enable the rapid development of the ever-broadening range of nucleic acid therapies to treat diseases that have no current cure. In particular, the 95% of rare diseases that have no FDA-approved treatment. And gene therapy is likely to be the answer to this, and our technology should help make that a reality.
Next slide, please. So when a manufacturer or a researcher wants to generate DNA, they first need to be synthesized. So de novo synthesis can be high throughput and automated, but only makes very little of the template DNA. They therefore need to make a lot more of the DNA template to administer a therapeutic dose or to test their product properly. So the method for manufacturing DNA has not changed substantially since the 1970s, hence this beautiful picture, and it has a number of serious limitations. For anyone who's never worked with DNA manufacture, it's expensive, it's time consuming, and a scientist in the lab might spend a couple of weeks just preparing and growing the cells, and 8 to 48 hours to extract the DNA, after all of which they might find themselves with very low yields. Next slide, please. Oh, sorry.
So E. coli cells want to make more plasmid unless they are forced to. This usually means adding antibiotics and an antibiotic-resistant sequence to the DNA. This should be removed before transfecting the patient, or it can reduce efficacy and safety and upset the regulators. There isn't sufficient supply to meet demand. Suppliers will therefore prioritize the bigger players, large-scale pharma, which leaves academia and smaller companies waiting for months on each order, with no guarantee on exactly how much they will get, and sometimes a high error level. In processes like cell therapy or any personalized treatment where logistics are complicated, this adds to the overall difficulty and therefore cost, and slows down the design, build, test cycle. Because it uses lots of high energy equipment over multiple steps and sometimes requires separate facilities from other activities, it is costly both for the user and for the planet. Next slide, please.
Cell-free methods for producing DNA are closer to a simple chemical reaction. So they're therefore more predictable, and there's no host cell genome for your plasmids to interact with. So you're likely to get a low error rate. And because we have direct access to all the components, we can have a greater control of quality. Number two, just like taking out the middlemen, removing cells means that more of the energy goes into producing the final product. For this reason, cell-free is highly efficient. With a still semi-optimized process, we can get 20 gram per litre, which is massively higher than cell-based, within a couple of days. And number three, finally, even ignoring the more efficient process, it's much cheaper for the user and for the environment. So our technology will reduce the cost and time of DNA production for our customers, our potential customers in the future, by more than half. Next slide, please.
So we want to commercialize a standalone automated DNA production device that will be an integral part of future biomanufacturing and biotechnology labs. Such device can be used to speed up the design build test cycle for nucleic acid therapies and later for distributed, localized, and on-demand manufacture for clinical applications. The subject of our R&R project is the development of the purification part of this device. So it needs to consist of as few steps as possible and purification methods which are amenable to automation in a simple device and based on liquid flows alone. The following few slides summarize very briefly the challenge we hope to address with the R&R project. Next slide, please.
So we know that the DNA our process produces, sorry, that was the previous slide, is not exactly like the DNA produced by cell-based production. So it is a higher initial purity and concentration, different background buffers and small molecule content, and for now at least, degree of supercoiling. Next slide, please.
We already know that conventional purification kits perform poorly in purification our DNA. We have seen very low yields from our attempts at purification to date. Next slide, please.
We also know that this technology has a very broad range of, very broad potential. So we can potentially make lots of different types of DNA using this method. But this means that we might need different purification methods for each. For example, if we want to use it for therapeutic applications, it needs to be very pure, and we need to make sure that we separate it from other similar species that might be in the mix. Next slide, please.
So by gathering high throughput data on a range of purification resins, which rely on different purification properties, we are aiming to be able to improve the purification of our DNA and to have the flexibility to pivot to different applications. Next slide, please.
So this little timeline here is just illustrating how we got to where we currently are, at least within the Research and Residence project. So about a year ago, we were attending a similar launch event to this one that we're currently at. And subsequently went to our PIs, the two of us, with 3 ideas for potential projects. We then contacted CPI, that's Central Process Innovation, and Cell and Gene Therapy Catapult. Cell and Gene Therapy got back to us and said they concentrate more on viral vectors than plasmids, therefore thank you but no thank you. However, CPI were interested in working with us. So I should say we had previously had a sort of some superficial work with CPI before. We'd done a couple of collaborative paper with them, but this was aiming to sort of deepen that relationship and get us access to the actual equipment and facilities, knowledge, etc. So we put together an expression of interest, which was refined in collaboration with CPI, our TRO and our PIs, of course, and were invited to full application. So we had the third, sort of the next development steps with CPI involved a telecom where we narrowed down our 3 ideas to 2, and then we went up in person to sort of present the 2 possible scopes for projects involving the 2 remaining ideas, and they helped us narrow down to the sort of one that they thought they were best placed to support us with and that they thought would have the biggest impact commercially. So this was very much based on their input. I suppose it's probably worth also stating that because our project is aimed at commercialization, we knew from the start that we needed to be thinking about IP, and if we were developing IP in collaboration with Catapult, how that might complicate later stages. So the arrangement we came to with CPI was that we would be taking essentially control of all the sort of say creative elements of the project, so development of the ideas and the approach, and CPI will be essentially providing facilities and generic advice based on their expertise. So just, I guess, to make people aware if that's an issue in your particular case. So CPI were able to sort of review our draft and help to sort of refine it, but the main core of the project was based on our own design. We then so obviously it went through, but I guess the other thing that is probably worth highlighting on this timeline. So we initially thought that we would be able, having received the go-ahead in March, to be able to start the project in July. It turns out, and again, this may be sort of old news to people that are sort of long-term academics, unlike ourselves, that putting together a collaboration agreement is quite a lengthy process. In our case, not because we were working with more than one catapult, but because things on our side at UCL were quite slow. So we did finally manage to finalise collaboration agreement only in December, and a couple of weeks ago attended our first week at CPI, which I think we would say was a good experience. We had our own we had our own Dutch initial training. People were very enthusiastic, happy to meet us.
It's an amazing environment, and we are willing to continue the project. We have access to everything. They are very low profile when it comes to discuss ideas and all the work. So it was very a fruitful week.
And I think we already learned a little bit more so that we understand how we need to adapt our project already to make it more realistic with the material and equipment that's there. And I think that's more or less everything we had to say
for now.
Thank you to Beatrice and Salome for providing that insight. You can read more about their project on innovationlaunchpad.ac.uk. Hopefully you found that interesting. Whether you're thinking about applying to our scheme, you know someone who might be interested, or you're just generally interested in what we're doing, we appreciate you all the same. As always, keep up to date with our activities on our socials, which is Innovation Launchpad Network Plus on Twitter/X, LinkedIn and YouTube. Our website is updated daily too on innovationlaunchpad.ac.uk, where you can find more about the projects from Tranche 1 and Tranche 2 of the scheme. Thanks again for joining us, and we'll be back next month with another podcast. Cheerio.